In this brief discussion, we'll talk a little bit more about transcriptomes and their predecessors, and we'll look a little bit at some of the things that are done in the process of creating transcriptomes. So originally, the first way that we got a snapshot of gene expression profiles in a large scale was with a microarray, technically a DNA microarray. But these, in the process of transcriptome analysis, would start with RNA samples. And these were made originally individually and later were made commercially and would represent a certain profile. So for instance, you could get a chip that represents every human genome. And every square on this little tiny piece of glass in the middle of this chip has been hybridized with a different human gene. And these were originally created by robots, little automated machines that would spot out a little sample of each gene onto each little different part of this plate. And so you could get ones that were entire genomes, you could get one that were specific for certain processes like cancer or heart disease or others. You get them for many different organisms, but primarily for humans and other major model organisms. And so what you would do is you would take two identical chips, you would put a sample on one and then a sample on the other, and then you would look at differences of expression profiles. And so the colors represent different levels of expression, and you can see that some genes are expressed in a tumor. They're different than what's expressed in a healthy cell. So this is very useful, and it's still used today, although to some extent RNA-seq has taken the place of this as sequencing of nucleic acids has become quicker and cheaper. Many of the big picture things that you can do or that are much more difficult to do with a microarray, we can do them now with RNA-seq. And that's particularly true for non-model organisms. So we didn't have microarrays available for a vast range of organisms unless you wanted to put all of the huge amounts of time and effort and money into making your own. You were stuck with looking at model organisms. And so microarrays are not obsolete, but they're becoming less used and maybe for more specialized processes like screening, quick medical tests, and other things. And RNA-seq to some extent has taken the place of DNA microarrays. And the most common methodology for generating sequences for RNA-seq for transcriptomes is Illumina's process. And so I've got a link here. It's a 5-minute video. You don't need to memorize all the details of it. I'm going to highlight a couple in the next slide that you need to be aware of. But you don't need to worry about all of the details. But I still think it's useful, especially if you're interested in it at all, or if potentially you might have a project that could benefit from this, or might be reading papers or referencing papers that benefit. It's useful to know kind of the state-of-the-art technology and what's going on. And it relies also on chips. But instead of having an array where different genes of known type are all put into a very structured arrangement, basically what we do is we take a huge lawn of these adapters. These little lines of balls represent nucleotides, A's, T's, C's, and G's. And these little things will bind samples that you put on there. And so you have to be very careful when you calculate the amount so that we don't overburden or waste your resources and not put enough. But the idea is to hybridize certain your samples to this chip, all of these little molecules that are on this chip. And once they're hybridized, essentially we can watch the synthesis process occur. It's done through chemistry and the emission of light as different base pairs are incorporated. And so what you end up with is a little bit like a random microarray with little lights lighting up for each of the 4 different nucleotides. And so for any one position where we have bound a sample, if we watch what lights are being activated during the synthesis of this strand, then we know what is there. Now, to get a strong enough light symbol, you actually have to amplify it. So instead of just a single strand, you have a whole bunch of similar strands clustered together. And again, you have to be careful that when you do this, they're not too close to one another, or else the software that's reading these flashes of lights is unable to discern between one patch of sequences and the other. It's a little bit more detailed in this video. You don't need to know all of the details. As long as you know kind of that level that I just explained, you're good. And then one other major thing I want to point out is that these methodologies generate large amounts of data. And so we had talked in the last lecture about how much coverage do I need to have, good coverage of my transcriptome. And it's not as much as a genome, and it's not based on any one particular size. Usually they do it based on reads. And so somewhere between 5 to 200 million reads for every sample is about what you want to have. Now, if you wanted to have better power to be able to detect rare transcripts, and you'd want to be more towards the 200. If you're just doing some basic genotyping, you can be down at the 500. And so what that means is that on one of these chips, you often have billions and billions of reads. And so if you only need a couple 100 million reads for each sample, then potentially what you could do is you could put on multiple samples on a single chip. But to be able to do that, we need to be able to somehow differentiate one sample from the other. And so to do that, we use adapters. Adapters are short oligonucleotides, a little bit like a primer, but a little shorter. They're short DNA sequences of known order that we can attach to our samples. So we could attach adapter 1 to the first sample, adapter 2 to the second sample, and so on. Then they all get mixed together on the sequencing plate. And then later, when we look at the sequences, all we do is say, hey, every sample that has this adapter on it, we're going to recognize it as from sample 1, and we'll put it in a separate folder, right? So we're looking at the analysis of these sequences on the computer afterwards. And if it has adapter number 2, it's from this other sample. And so we can separate out the different samples post-sequencing and then analyze them separately. So it's really useful, a nice way to get the most bang for your buck. So you can do maybe 10, 20, depending on the depth of sequencing you need, maybe even dozens and dozens of samples on a single run. And the end result are paired reads. Now, a paired read is basically still a randomized chunk of your target, which in this case is a transcript, but the paired reads have a relatively consistent amount on either side that is sequenced, and then a fairly, there's a range here, but a fairly consistent gap in between them. So we don't know the sequence here for the blue line, but we do know the sequence for the two reds. And it turns out that the algorithm for assembling sequences is actually a little simpler and a little easier if we have paired reads, because we have some additional information. Not only are these random reads, but we know about how far apart they should be. And so it makes assembly a little bit easier. But notice that you still have areas where you might have only 2 or 3 sampled sequences for that part. But in other areas, you might have many, 5, 6, 7, 8, or more. Again, depending on the depth of sequence, you're going to be sampling some parts over and over again. So once you've generated these reads, before you assemble them, there's usually a quality control process. We'll talk in some vague detail about this in the next discussion when we go over differential expression. Okay, but you do a quality control, which ones are really good reads we're certain of and which ones maybe we're less certain of. And so we may be able to weed out some of the sequences that might be of lower quality, or perhaps recognize and remove adapters or other contaminating sequences that might have carried over into your final product. And then, of course, we do an alignment. Now, if we are aligning a transcriptome from an organism that we already know very well, then we will often align them to a reference transcriptome, just like we did with the reference genome when we talked about that. So if you're doing a fruit fly or a human or Arabidopsis, C. elegans, any of these model organisms for which we know the transcriptome pretty much completely, right? We know every gene in their genome. You can align them much more easily, and it's easy to identify genes as long as we have good quality sequence and map each read to a very specific gene. However, if it is a new organism that has never been worked with before, then we need to do a de novo one. And this can be a little bit tricky, particularly if we have areas that are very similar between, for instance, gene A and gene B. If they are duplicate genes of one another, then we might have very, very similar regions. And so it can be a little tricky. De novo, it takes a little bit longer, and you have to be a little bit, the analysis is a little bit different. Okay, but both of them use these paired reads. Okay, once we have assembled our transcriptome, then we can begin to work with it. The very first thing is simple quantification, which basically means we get a relative estimate for how many times we have gotten a sequence for a particular target. And these can be called transcriptomes or genes or even identified gene products, right? So we quantify. And then if we have more than one transcriptome, we can do some differential expression analysis. So gene 1 is expressed at a certain level in transcriptome 1. Maybe that transcriptome 1 represents a liver cell. And then we can look at that same gene and its level of transcriptome in sample 2, which might represent a nerve cell, for instance. And so by analyzing that, we can tell if they are significantly different. And if they're very, very highly different, then that is one of maybe many genes, but at least one of the genes that is critical in the different performance of those cells. So differential expression can be really, really powerful. And the Lau paper looks at the one that was the reading assignment for Monday, November 2nd, looks at the end of that and lists some of the things and gives you some general ideas about the different things. We've talked about some of them already in previous lectures. And then finally, often, particularly if you're drawing some pretty strong conclusions from your differential expression analysis, often there will be some sort of independent validation or verification of these expression levels. And the most common way to do that is qPCR, or quantitative PCR. This is a diagram showing the output of a qPCR run. Basically, what you do is you look for when a sample becomes amplified. And depending on when that maximum amplification is reached, that you can extrapolate back to determine how much you started with. So things that amplify rapidly, we had a large number of copies to start with. Things that amplify more slowly, then we had fewer copies. And so if I've got a gene that I'm saying is expressed at X levels, I could validate that independently using qPCR. And if I get similar values, then I'm very confident in my original estimation based on my differential expression analysis. And then the final way, these are more big picture ways that we can look at entire transcriptomes. And I'm going to talk about these in more detail. For now, I'll just list them and briefly outline them. And in Wednesday's discussion, I'll give you more details about exactly what these are and what they mean and how they work. And we'll even look at some examples from the literature. Okay? But a heat map is basically a comparison across samples and across different genes and gene sets to see which samples are similar, have similar expression profiles, like samples 4, 5, 6, 7, 8, 9 have roughly similar. Some are overexpressed in red, some are underexpressed in blue, but they're roughly similar, where samples 1, 2, and 3 have very different expression profiles from the rest of them. So we can start to get a big picture idea, and there's a ton of data and information here based on how dark the color is and the color of what color it is. We can look at overall expression patterns across a wide sample of alleles. So a wide sample of transcripts. So it's useful to give you a big picture one, but can be a little bit overwhelming. And then finally, a volcano plot is simply a comparison between two different samples and looking at genes that are statistically overexpressed in red in whatever your reference sample is, and genes that are underexpressed. Now realize these are comparisons. So when I say overexpressed, I'm talking about in reference to one of the samples. For every gene that is overexpressed in sample A, that means it's going to be quote unquote underexpressed in sample B. So the red ones represent genes that are differentially expressed up in one and down in the other. And if we did the comparison the other way, it would just simply flip the volcano plot along the axis here, and you'd have the same thing just in reverse. Okay, and some are very, very highly expressed. Some are just barely. The ones in gray didn't reach a critical level of significance as far as expression. And so often what people will do, particularly if they're looking for a candidate gene or a candidate set of genes that are important in a function, is they will start by analyzing these ones that are most underexpressed or most overexpressed as key indicators and key factors for what makes those cells different from one another.